Good day everyone, and today's session we will record something that lies at the very core of life, DNA. Whether you are preparing for the SST biochemistry exam, aiming for MDCate, or simply want to master molecular biology, this lecture is tailored to give you conceptual clarity and MCQs confidence. So let's dive in and stay with me till the end, because I will be testing your understanding with high-level exam questions and explanations. So let's start with what is DNA. DNA, short for deoxyribonucleic acid, is a genetic blueprint for life. It carries the instructions required for the structure, function, and regulation of your cells, tissues, and entire body. Now the MCQ that can be asked: which of the following best defines DNA from a molecular biology perspective? So a protein coding RNA molecule. So no, it's not going to make a sense because it's a DNA molecule, not an RNA molecule. The next option is a double-stranded polypeptide chain. So yes, it's a double-stranded molecule, but it's made up of nucleotide, and polypeptide chain is something that is made up of amino acid. So it's not going to make sense. The next is a nucleic acid that encodes heritable genetic information. So yes, right, it's going to make sense because it's a nucleic acid and it's also a hereditary material. The next is a cytoplasmic regulatory element. So no, it's not the right answer. So the right option is the C option. So DNA is a type of nucleic acid. Its function is to store genetic information and pass it from one generation to the next. It's not a polypeptide or protein. It's the code that tells the cell how to make proteins. Now, where is DNA found? In eukaryotic organisms, DNA is located inside the nucleus, carefully packaged into chromosomes. In prokaryotes, it's found in the cytoplasm, since prokaryotes lack a defined nucleus. Now the MCQs that can be asked by the examiners are the which part of a eukaryotic cell contains transcriptionally active DNA. Now let me share the structure of the chromosome. This is a chromosome structure. Now this portion is known as a telomere portion. This is the centromere portion, right? The loosely packed chromatin called euchromatin. Now the options are the given as the telomeres, nucleus, euchromatin, and centrioles. So no telomeres is not containing transcriptionally active DNA. Nucleolus also don't contain transcriptionally active DNA. Euchromatin. So yes, it's going to make sense. Centriole is something that is present in the animal cells but not actually related to transcriptional activation of DNA. So the right answer is the euchromatin. Euchromatin is loosely packed chromatin that is actively being transcribed into RNA. Telomeres are protective caps, not transcription sites. So the DNA have unique structure. DNA has a double helical structure like a twisted ladder. This structure is perfect for storing information compactly and making accurate copies during cell division. Now the MCQ can be asked as what enables DNA to serve as a reliable template during replication. So it's enzymatic activity. So no, it's not going to make sense. complementary base pairing. Yes, DNA have complementary base pairing, and it's also have role in the during replication. Covalent bonding between bases. So there is no covalent bonding between bases. Between the bases there's a hydrogen bond. Supercoiling, so it's not something for the related to replication. Supercoiling is related to the activation of, is a more compact version of the genes. So DNA strands pair specifically A with T and G with C, allowing the cell to easily create an exact copy of the strain during replication. So the right answer is the complementary base pairing. Now what does DNA stand for? DNA stands for deoxyribonucleic acid. It's made up of monomers called nucleotides, and each nucleotide contains a sugar, specifically deoxyribose sugar, a phosphate group, and a nitrogenous base. Now in the previous past paper there was a question that which of the following is not found in the DNA. So the option was given as a deoxyribose, phosphate group, nitrogenous base, and ribose. So the right answer was the ribose, because ribose is present in the mRNA. Ribose is the part of the RNA, not the part of the DNA. DNA have sugar deoxyribose. Now the MCQ question that can be asked by the examiner is, what makes deoxyribose different from ribose in RNA? So deoxy and DNA means one oxygen is missing, specifically on the second carbon of the sugar. So this difference contributes to DNA stability. So the right option is the absence of oxygen on the second carbon and deoxyribose. And the other options is not going to make sense. Now the double helix model of DNA was proposed by Watson and Crick in 1953. It's stabilized by hydrogen bonds between nitrogenous base and has a right-handed twist. The MCQ question can be asked here as, which technique revealed the helical structure of DNA? So it was X-ray diffraction. Rosalind Franklin X-ray diffraction image shows that X pattern typically alpha helix structure, helping Watson and Crick build their model. So the X-ray diffraction image, which showed by the Rosalind Franklin, helped in the discovery of the DNA structure. Now this video is incomplete without mentioning nitrogenous bases here. The rungs of the DNA ladder are formed by nitrogenous bases A, T, G, and C. A stands for adenine, T stands for thymine, G stands for guanine, and C is for cytosine. These bases pair in a very specific way, which leads us to our next concept. Now the MCQ can be asked as, which of the following base pair is held together by 3 hydrogen bonds? Now the first option is A and T. So adenine pair with thymine with double hydrogen bonds, not with triple hydrogen bonds. G and C, guanine and cytosine, they pair with triple bonds. C and T, it's not going to make sense because C never pair with T. A pair with G, no, it's not also false because A never pair with G. A pair with T but with double bond. G pair with C, and this is the right option because G and C have triple bonds. So guanine pairs with cytosines using 3 hydrogen bonds, making it stronger than A pair, which forms only 2 bonds. Now the backbone of the DNA is the sides of the DNA ladder are composed of alternating sugar and phosphate groups joined by strong covalent phosphodiester bonds. Now the MCQ question can be asked as which type of bond links nucleotides and the DNA backbone. So the right answer will be phosphodiester bond, because phosphodiester bonds connect the third carbon of one sugar to the fifth phosphate of the next nucleotide, forming the sugar-phosphate backbone. Now the base pairing rule is an DNA base pairing followed a fixed rule. A pairs with T, Y2 hydrogen bonds and G pairs with C, Y3 hydrogen bonds. This ensures consistency during replication. Now a tricky MCQ question that can be asked in an exam is if a DNA strand has 28% adenine, how much guanine does it have? So it's really tricky. If DNA strand has 28% adenine, so there will be 28% thymine, because adenine pairs with thymine. So 28 plus 28, this will make 56. So 56% of the total. So the 56, 100 minus 56, so the remaining will be 44%. Right? So the 44% is shared equally by G and C, because G and C paired with each other. So 44 divided by 2. So the right answer will be 22%. And here is the 22%. Right? So this is a very tricky question that mostly can be asked by examiners in the exams. Now, how does DNA work? DNA controls cell function by using codons, sequence of 3 bases that code for specific amino acid. 3 nucleotides combines together to form a codons, and these one codon code for specific amino acid. Multiple codons can also code for a specific amino acid. And now these codons form genes, and genes instruct the cell on how to build proteins. Now the MCQ question here can be asked as the process of converting DNA to mRNA is called. The options are given: translation, transcription, replication, reverse transcription. So the right answer is the transcription, because transcription is when DNA is used as a template to make mRNA. Translation is the next step, turning mRNA into protein. Now let's dive into the DNA discovery. Watson and Crick discovery of the double helix structure earned them a Nobel Prize in 1962. But it was Rosalind Franklin X-ray data that revealed DNA shape, a contribution sadly overlooked at that time. Now the MCQ question that can be asked as who experimentally proved DNA is a genetic material using bacteriophage. The options are Avery, MacLeod and McCarthy. So no, it's not the right option, because they take the S strain of the bacteriophage and treat it with the different digestive enzymes like DNase, proteinase, RNase, etc. So it was not about the proving of DNA as a genetic material. Hershey and Chase experiment. So yes, it was something like Hershey and Chase used radioactive labels to show that only DNA, not protein, entered bacterial cell, proving DNA carried genetic information. So Hershey and Chase, this option is going to make sense. Watson and Crick Watson and Crick discovered the double helix structure of the DNA, not proof it as a genetic material. Griffith experiment was about the S strand and R strand, the transfer of the virulency from in the bacteriophages generation. So it was not about the proving DNA as genetic material. It was all research. Every nucleotide and MCQ, it's this A and D. This both experiment was discovery and progress towards DNA as genetic material proof, but it was not specifically proof of the. But they don't specifically prove that DNA is a genetic material. So the right answer is the Hershey and Chase. Now that brings us to the end of this lecture. Now I am going to ask you a tricky question. Here's a question to leave you thinking. If DNA holds all the information, then why does not the cell just use DNA directly for protein synthesis? Why take the extra step of transcribing DNA into mRNA? This might seem like an inefficient detour, but it is one of the smartest strategies nature has evolved. We will decode that in the next lecture when we dive into transcription, RNA types, and the beautiful process of translation. Until then, let this question stay with you. If you have followed along until now, you have just reviewed 10 of the most exam-relevant concept-rich MCQs, not just memorized facts, but actually understood the why behind the answer. Thank you for watching, and remember, the better you understand DNA, the better you understand life.